You're going to see some kind of overlap with this in terms of climate change, ocean acidification, ocean warming. I should also point out that this is part of a much larger project that has yet to be finished with the genetics portion of this. I am strictly focusing on the physiological, as that's my role in the lab. And this is going to take form in a species analysis, which has been kind of thrown out in the later portions of this lab, just because we didn't get good genetic tissue on one of the species. So we changed it from a species analysis to a single species study. But I said, heck with all that, we still have the physiological data for this, so I still want to do my species analysis. So this is going to be physiological markers associated with byssal attachment following environmental perturbations in marine mussels. And to get you started out with some background information, the global bivalve aquaculture value is at about $20.6 billion per year as of 2020, I believe. And mussels make up 13% of the global market in U.S. seafood trade. So all those mussels are getting exported to primarily Canada, Spain, and other European countries. And with this, the big problem is that this creates a seafood deficit because in the U.S., we love our cheap food and we love our cheap seafood. So we actually import a lot of seafood into the States from Asia and other aquaculture facilities over there because it's just cheaper to import it over there than buy our own. So the deficit is about double the US per year aquaculture value at around $20 billion per year, and that's looking like it's going to be increasing. So starting with the biology of mussels, we have the byssal threads, which are protein-based, unlike oysters, which produce kind of a cement to stick them on. They have kind of these tendrils that come out and hug onto rocks. And these byssal threads are dopa-rich mussels. So if you see MFPs, it just means mussel foot proteins. And those are those tendrils that are critical for holding onto those rocks and survival growth and to increase aquaculture yield. Because, as for the next slide, climate change versus mussels, we have ocean acidification, which is the acidification of ocean water, particularly kind of an important subject in the Sound since we're kind of a more closed in space. Especially important for aquaculture, which are suspended in kind of cove areas. We have ocean warming, and over the last 40 years we've seen about a 0.9 degrees Celsius increase. And hypoxia, which is our final metric that I've measured, which is coastal oxygen declines. So eutrophication, warming, algal blooms, dead zones, if you've heard all that, that's what that is. So trying to solve the problem is that we want to investigate byssal thread production and metabolic performance within the two species Mytilus trossulus and Galloprovincialis. So these are our two mussel groups. So trossulus being the Pacific blue mussel, which is native to the Pacific Northwest from the Sound all the way up to Alaska, and Galloprovincialis, which is the Mediterranean mussel, a highly invasive mussel that is produced in aquaculture here. So we're going to be examining the stress effects of ocean acidification, ocean warming, and hypoxia with these two species. So some of the stuff that we already know is ocean acidification and ocean warming are the primary, focus on a lot of previous studies. And they have shown to have reduced thread production, but they haven't focused on specific aquaculture species that are important to the Pacific Northwest. A lot of it's to do with the blue mussel, which is different from the Pacific blue mussel. So a lot of previous research on other mussels, but not ones that relate to us and our economic value here in the state. But some studies also show some adaptive potential with unregulated proteins, so that shows some plasticity to when they are exposed to heat or acidification. They actually release more of those tendrils or produce more of those proteins to adhere to the rock better. So, taking all the previous research and taking all of the ideas of what we want to, how we want to go forward with this, is we came up with the hypothesis of stress, effects, thread composition, structure, and performance. So, this is just focusing on the threads, but it is also important that we're going to be looking at SMR, which is the systematic metabolic rate, as we'll see a little bit later on. But for now, we're going to get into some of the experimental design, and how we actually went about this is that we took 200 mussels per species, so 200 Trochelus and 200 Galloprovincialis. We got them from Penn Shellfish Cove off of the coast of Whidbey Island, and Galloprovincialis also from Penn Shellfish Cove, but their farm for the Mediterranean mussels was in Hood Canal. But here's our 4 treatments. We have our control, acidification, warming, and hypoxia. These will pop up in a much easier to read results section later, so don't worry about that. But the setup, we had mussels placed on mica plates, so it was an easy mineral for them to adhere to, and also clear, so we can take photographs and measure the length of the abyssal threads. And then we have the microcosm tanks, which is basically just a small ecosystem that keeps them relatively comfortable so they aren't stressed out by outside factors. And then we counted threads before and after the stress exposures to get an accurate count, as well as for the SMR ratings. Here is our definitely not illegal setup that looks like some sort of drug lab. And then we have our oysters, and I don't know if you can really see, but it's like a clear plate. That's what the mica is. And then for respirometry, we did a closed system and measured oxygen uptake through milligrams of O2. And we did pre and post treatments controlled for temperature, salinity, and a point in chamber. This is very similar to kind of our thread count, just changed a little bit. We have the same controls. And then these are the statistics that I used for R. So I spent the better part of my last summer writing R script and trying to run all of these. I think there was about 2,000 thread samples that I had to kind of synthesize, analyze, and then normalize. So there was some ambiguity in some of the photos, so we just had to take some of those out. But for the statistics through R, we used an ANOVA and a COVA for repeated measures, and then we did some normality checks with QQ plots. So some of our predictions are that we're going to be able to identify physiological markers for OA and OW resilience. So trying to see some of the adaptability or the plasticity of each species. And the big goal, since this is funded partially, and we were given mussels by Penn Cove Shellfish Company, is that we can give them insight for how to improve mussel farming and sustainability. Also, larger picture is that seafood trade deficit of all of our canned seafood and mussels and stuff like that being from foreign countries on our shelves, we tried to create a better aquaculture environment here in the States and offset that seafood trade deficit. And then, of course, physiological markers enhancing industry resilience, so not just for us, but the rest of the world and aquaculture. All right, finally get to the results. We have ocean acidification, warming, and hypoxia. So we can see that we start out with Galliporencialis slightly lower in a median, and they both jump up a little bit in threat number in response to ocean acidification. Not much of a difference, and they stay relatively equal, at least with their medians. And then you have ocean warming, which kind of has the same trend, but they swap places with Galliporencialis actually producing more threads in a ocean warming environment than Trosylus, which we are hypothesizing that it has maybe something to do with the Mediterranean mussel being from a warmer climate, being able to adapt or have that plasticity compared to our native Trosylus blue, Pacific blue mussel variety. For hypoxia, this one was actually kind of interesting because you notice that Galliporencialis is nowhere to be seen on this, and that is because they produced no threads. They just kind of, we got back to them and they were kind of just free floating in there. And we tried it again, same deal, they just didn't want to produce threads for us. They were still alive, just didn't want to produce threads, which Trosylus also exhibited kind of a steep decline. So round one with thread number is that hypoxia seems to be the most concerning out of the three climate change variables that we were looking at. And now we're going to move on to the metabolic rate, where we have ocean acidification with actually the Pacific blue mussel having a higher respiration or metabolic rate, so really working through that energy faster than the other. And Galliporencialis actually taking a decline and reducing its metabolic rate, which is interesting for a number of reasons. That in acidification, it kind of shuts down or slows down its metabolism. And then we have ocean warming, where they both, this is the most unsurprising one, is that the medians for both species increase with warming, because metabolic rate, you increase warming, you increase your energy processing times. So this one wasn't really too surprising to see. And then finally, hypoxia skyrocketed for both Galliporencialis and Trosylus. Hypoxia, they were essentially hyperventilating in their little chamber, which felt bad for them. Why is this important for aquaculture is that we want to understand climate resilience, and it's extremely important for successful harvests here in Washington. Site selection should definitely be considered for species to species tolerance. I know that there's talk more in near the border in Blaine about some out of water aquaculture rearing for mussels and oysters in actual tanks and not suspended raft culture like these were. And we also saw that Galliporencialis might be more robust under ocean acidification, ocean warming, and hypoxic conditions, which kind of gives, thinking back on, is not as surprising since they are a extremely invasive species here in the Washington state area. But the key takeaway, I think, is that hypoxia remains the major stressor for both these mussels, and dead zones and eutrophication is one of the main things that we need to be kind of worried about in some of the canals. Some limitations and future recommendations kind of for myself or future endeavors or projects that run along the same line. is that we only did 3 days of acute exposure. So the chronic stress, we couldn't really look at. We just kind of did them for 3 days, tried to see what kind of change they had. But definitely a longer response period would be nice to see. And then mechanistic studies on the muscle foot proteins. So actually dissecting them, looking at the chemistry and seeing if there's anything fundamental that changes. And then applications to selective breeding and site management. So we didn't take into account the actual Penn Cove shellfish and what their breeding strategies are, or how they've messed with their site selection or selective breeding. For key takeaways, so big picture stuff is species specifics are significant. So it's not just a one-size-fits-all problem. And climate change stressors impact muscle physiology. So we saw thread numbers go up in certain areas, some plasticity, some ability to adapt, and then, for example, the hypoxic environments, it just kind of takes them all away. Aquaculture should leverage physiological insight and practice resilience-based farming. So this is more of that kind of selective breeding and kind of getting their sites down to where they choose sites to rear aquaculture facilities that don't show as much of an impact to climate change. And I would like to just say thank you to the team. This is Stephen Roberts. And then my worksite supervisor, aka the postdoc that I was assigned to, Matt George. Thank you to them, and thank you to the Penn Cove Shellfish Company for supplying the mussels. And thank you for listening. Let me know if you have any questions. This is kind of the big project timeline there.
I may have already forgotten, missed it, but for the hypoxia study, was it like full term, like from the start of the experiment to the end of the experiment, hypoxia, or did you play with any like transient hypoxia conditions?
It was full, just the only one that we did a ramp for is the ocean warming right here, where we kind of increased it by 2 degrees Celsius over time. But for all the other ones with hypoxia and ocean acidification, it was just straight in there.
It's interesting with Galliprovincialis not responding with distal threads during the hypoxic event. I wonder if there would be like an on and off growth, reduction of that distal threads during transient, like short-term, periodic on-off hypoxic events. It's pretty interesting.
Isn't there an issue with those two species hybridizing to some extent?
I haven't looked into that at all, because that's an interesting point, because I haven't looked into any sort of hybridization. That would be interesting to look into, and look into if it would, I guess, make them a little bit more robust, but we also wouldn't want that into like wild populations. But I have no clue on hybridization.
Yeah, did you, so you're talking about how they weren't in these like, containers, they're out in the ocean.
Yes.
Did you kind of like, or at least Penn Cove, think of any like sort of husbandry changes that could combat the hypoxia in particular if it's not a contained system?
Yeah, so how their raft culture kind of works is that they have specific breeding groups. So they have like a giant whiteboard, and they have different plots or different squares of raft culture. And those are kind of a family tree of which ones are doing the best, which ones are kind of producing the most, and those are the ones that they want to seed for future raft culture mussels.